So, hi everyone. So in this video, we're going to continue with subtopic 6.4, gene regulation and expression of like operon. We have learned gene expression for eukaryotic in previous video, and in this video, we're going to talk about gene expression in prokaryotes such as bacteria. The learning outcome for this topic is at the end of the lesson, you should be able to explain the concept of operon and gene regulation. So let's get started. You know that in each cell in the human contains all genetic material that was stored in the nucleus, and in the nucleus you have DNA, and this DNA is what we use for growth and development of a human. So how does this happen? So it is by the transcription and the translation of DNA and mRNA to a functional protein, or this is what we call as gene expression. However, you need to know that not all genes are expressed all the time. Only some of these genes will need to be expressed throughout the time. For example, genes that are involved in vital biochemical process such as respiration. This kind of gene is need to be expressed all the time because it is used for respiration where you do respire all the time. So, for gene expression in prokaryote, it can be explained by the Operon model. So, this Operon model describes the mechanism for the control of gene expression in bacteria, such as E. coli. So, this part here is a segment of bacterial chromosome, and this encodes for several genes. These genes that work together will be transcribed into mRNA, and then it will be translated into protein. Note that some of the bacterial genes will be expressed only when it is necessary. So there's one fun fact. The DNA of E. coli is sufficient to encode about 4,000 proteins, but only a fraction of these are made at one time. So E. coli regulates the expression of many its genes according to the food sources that are available to it. The Operon model was proposed by Francois Jacob and Jacques Monod in 1961, and their work is on E. coli. So the E. coli-like operon is an example of an inducible set of genes. Transcription and regulation of certain genes is needed to break down lactose into sugars. So this is used for cellular metabolism in E. coli. You will learn this more in the next video. Okay, for your information, E. coli grow rapidly on a culture medium containing glucose. But when E. coli is transferred to a medium containing lactose, it will show a short delay to grow. This is because the growth in lactose required a presence of enzyme, in which this enzyme is needed in order to hydrolyze lactose to glucose and galactose. You have learned this in chapter 1. So, lactose has induced the synthesis of particular enzymes. So when these enzymes are present, then only the E. coli can utilize lactose for cellular metabolism. So, whenever glucose is present, E. coli will metabolize glucose first, before it uses alternative energy sources such as lactose. This is because glucose is the preferred and more frequently available energy for E. coli. The enzyme to metabolize glucose are made constantly by E. coli. That is why when you grow E. coli in a glucose-containing medium, it grows rapidly. Next, an operant model of prokaryotic gene regulation. You have groups of genes encoding for related proteins that are arranged in a unit known as operon. This is how the operon looks like. In an operon, it consists of an operator, a promoter, and also a structural genes. This component is involved in genes expression for prokaryote, so it will involve for the transcription and also translation until it produce or get a functional protein. So this is all for this part of the video. Thank you.